every single day we discover something new because technology has evolved to help us develop drugs in a faster way and in a more precise way. We are really at a position that hope is everywhere.
Hello and welcome to Washington Post Live. I'm Frances Stead Sellers, an associate editor here at the Washington Post. Today we have two segments on the future of cancer care. In a little while, I'll be joined by Professor Georgina Long from the Melanoma Institute, Australia. But first, I'm delighted to welcome Dr. Samir Khalif. He is the founder and CEO of Georgia Immune, a US-based biotech startup. Samir, a very warm welcome to Washington Post Live.
Thank you very much, Frances. Great to see you.
Thanks for joining us. So I wanted to start with the big picture. Let's talk about the numbers. In the last 2.5 decades, we've seen a big drop in mortality from cancer, but some 600,000 Americans are still dying every year. So can you set the scene a little bit for us and also tell us what immunotherapy offers that traditional therapies do not?
Yeah, so you're absolutely correct. There is a major decrease in mortality in cancer for the past many years. And that, of course, was helped by the Cancer Act in 1972 and the tremendous amount of funding that went into research and cancer research. The issue is, unfortunately, we've made a tremendous amount of progress. However, as you mentioned, there's still many die because of advanced disease and because of limitation still of the therapies that we have currently available, particularly for patients with advanced cancer. So I'll pivot towards immunotherapy. So immunotherapy now, it's been in existence, at least in use for patient, general use for patient, for almost 10 to 12 years. And when immunotherapy came to being and became part of the cancer therapy repertoire, it changes what was already available, which is surgery, radiation therapy, and chemotherapy. And until then, we're talking about therapies that have been in existence for 100 years, chemotherapy for almost 70 years, without major progress. So when immunotherapy came to being because of major discoveries of understanding how the immune system works, and more importantly, how the cancer affect the immune system in a negative way, suppressing the immune system, and how to overcome that, this led to a major revolution in cancer care. So some patients that never had a chance before 2012 or 2013, that used to really have a lifespan of almost 6 months, now can be cured. That's the good news. Sorry.
So let me ask you a little bit about how you have tackled this. You were at the National Institutes of Health and then at the Georgia Cancer Center at Augusta University, and now you've switched to running a startup. Tell us about what prompted that switch and how it changes your approach to tackling the changes you're talking about.
Yeah, that's a great segue from the past question. Well, look, so I don't see it as a change. I'll see it as a progression, as continuation. And I still have an academic job, and I'm a professor at Georgetown. I still run research there at the university. Having said that, what I felt that to a certain degree, Francis, a lot of discoveries, unfortunately, in academia, as good as they may be, they are not progressing or do not progress into commercial use and into patients' benefits. The reason, because many universities are not equipped of taking a discovery and intellectual property and transfer it into a use to patients. That requires industry, that requires biotech, that requires funding. So the transition that I made is because of the multiple discoveries that we had while I was the director of the Cancer Center for University in Georgia. I decided to personally take the bull by the horn and form the company so we can take those intellectual property of the discoveries and the potential utilization of those towards development of drugs to benefit patients. Now, why? Because this is all new science. And the issue is, and what I was going to say in the previous question, is despite the fact of the progress of immunotherapy, only 15% of patients respond to immunotherapy, 15 to 20%. So we have a major bulk of patients, 80 to 85% of patients still don't respond to immunotherapy. Accordingly, they need new science. They need new type of drugs that are or can be developed so we can attack the cancer in those kind of patients. That's what I decided to do.
You've taken my next thought out of my mouth, but I was going to ask you exactly about that 85% and how you at Georgia Immune are trying to address the challenges facing that big bulk of the population who are suffering.
A great question. So, see, the issue, what helps me in that is the Nobel Prize that was announced 2 days ago, which was given to another discovery in immunology, which is the T regulatory cell, which is a type of cell that inhibits the immune system, and cancer take advantage of our cells in the body, our immune cells, and convert them, those cells that are coming to attack the cancer, it converts them into the T regulatory cells, so become inhibitors. Now, why am I telling you this? One, because I'm celebrating the Nobel Prize, because one of the people that got the Nobel Prize is a very close friend, and he's an advisor at our company. And the second is because we decided to move away from what already succeeded in immunotherapy. So the checkpoint inhibitors that were developed in the 2012, etc., that led to that revolution, that led to the response in 15% to 20% of patients, plateaued. We need something new. We need some new science. So what Georgia Immune is doing is discovering new science. For example, in T regulatory cell that got the Nobel Prize, we discovered the master switch of that T regulatory cell. Accordingly, we can turn it off in cancer patients. We can turn it on in other diseases that require it, but we can turn it off in cancer patients. So that's one of the new drugs that we've developed and now being tested in clinical trials to get away from the traditional things that succeeded into other part of science that we believe need to be explored. And we know it has a major role in cancer development and cancer spreading and the failure of responses. So that's one. So what Georgia Immune is doing is really developing very new science. We were able to develop now 10 drugs, all of them based on very novel science and very new biology that leads to the development of therapeutics that could target those 80% of patients. So currently we have 2 drugs for cancer and 1 for autoimmune disease, but 2 drugs already in clinic that were based on those new discoveries that tap into that. What I call immunology is like a tip of the iceberg. We know only the tip. There is major part under the water that we don't know. And this is what's going to help the 80% of patients. So we need to dig into that. And this is what Georgia Immune is doing.
So you are talking wonderfully about the science, but we know with innovation there are always inhibitions that come from cultural and economic barriers as well. Can you dive a little bit into that and tell us what's holding up innovation in this very promising area you're talking about?
So this is a topic that's dear and dear to my heart. And let me take that 15% success example.
Right.
Culturally, and you literally hit it on the nail. Culturally, in the community, when something succeeds, everybody follows that and they forget about everything else. So I was talking about the tip of the iceberg. So when the tip of the iceberg therapy succeeded, everybody in the community, whether pharma or funding, a lot of funding, or even investors, followed that path. And they forgot that there's 80% of patients that nature and God created immunology for them too, not only for those 15% of patients. So that herd mentality led to delay in progress. Maybe not only herd mentality, maybe also conservatism in the mentality of pharma sometimes, or some funding, like venture capitalists, etc. If this is what succeeded, maybe we should put all our effort in that, and why take a risk on other part? So I think all this combined from a cultural perspective, as you mentioned, Francis, led to slowdown of progress that we've seen 10 to 12 years ago. And hopefully that's changing now big time.
So help me understand a little better the sort of crossover between the personalization of medicine, these very targeted therapies, and our need to address what's become basically chronic disease and public health issues that are sweeping across this country. And can AI feed in? Are we learning technologies that can help do both things at the same time?
Yes. So AI is definitely one of the most promising advances in biology to understand the diseases better, but as importantly, to be able to develop drugs and make this process more accurate and faster. That's for sure. As a matter of fact, Georgia Immune, every single drug we developed in Georgia Immune has been AI assisted. And we implement AI in many ways. Of course, I'm not talking about ChatGPT or LLMs here. I'm talking more about modeling and structural modeling and protein modeling. AI is critical for that. So to get back directly to your question, so it can help us in two ways. One, discern those type of diseases, including chronic diseases, and understand more about them. And in the future, probably in the next 5 years, we'll be even more inquisitive in a way that we probably could understand the pathology or understand protein relevant to those pathologies. Because at this stage, we really have to do lots of work for years to be understanding those kind of proteins. I think that's one. And then the second, shrinking the time and probably make it more affordable, the development of drugs. As you very well know, to develop a drug takes almost 12 to 16 years and between $2 and $8 billion. So that is definitely going to be assisted by AI in a way that becomes more efficient and faster.
So I don't think we can talk about the future of cancer care without touching on this issue of mRNA vaccines and their potential to help. How would you see them in the ideal world fitting alongside immunotherapy and these advances in drug production that you're talking about?
Yeah. So in many ways, so vaccine is definitely, so I just wrote a review article for one of the major journals about cancer vaccine. And I called the article, the title of it, Cancer Vaccine, an Enabler to Immune Therapy. So what does vaccine do, right? What does vaccine do? So what vaccine does, it enhances the immune system. It trains particularly T cells and maybe sometimes B cells, particularly T cells. It enhances them and it trains them to recognize the antigens in cancer, specifically in cancer that are presented by cancer and try to attack the tumor. So which means that it creates soldiers. Now, those soldiers cannot work if the cancer has a defense mechanism around it that doesn't allow the soldiers to come in. And there are many of those defense mechanisms that cancer develop. And the immunotherapies that we're currently using in the clinic is to at least abolish or tear down a couple of mechanisms of those. But what vaccine can do is enhance the soldiers, is increase the T cells. So clearly it has a role in immunotherapy. And we've shown that if you give vaccine, it enhances the effect of the checkpoint inhibitors for many reasons. Actually, it might flip sometimes the resistance to those checkpoint inhibitors, which are currently in the market, and makes them work better, or sometimes make them work where they don't work. So there are many clinical trials now that are showing that vaccine would have potential role as a combination with other immunotherapeutic approaches. Now, one thing I would say though, Francis, I would not limit it to RNA vaccines. RNA vaccine is one type of vaccine, but there are other vaccines that could also be very effective and will generate and will lead to the same outcome. Now, we know RNA vaccine was very effective in COVID, and it has a lot of advantage to it. But I would not say only RNA vaccine. There are other vaccines that might be of value also in cancer vaccine approaches.
So when we heard this summer that the Department of Health and Human Services was halting some work on RNA vaccines, that's a halt in one part of this progress, but you're saying that other platforms or other vaccine platforms could work and advance here.
Yeah, it can. It absolutely can. And there are many platforms of vaccines, not only RNA vaccines, although again, RNA vaccines are effective. We cannot brush this off. But there are other type of vaccines that can be used in cancer approaches, in cancer vaccine approaches.
So give me some of your optimistic views for the future. What do you hope to see both on the research side and for patients if you're talking to them about what the future holds for these very troubling and frightening diseases?
Yeah. So I'm very optimistic by nature, right? I'm an oncologist. I have to be optimistic. And I'm a scientist. I have to be optimistic. So that combination, it has to be. If you're not optimistic, you should get out of this field, right? But I can tell you one thing. The advances in science in the past 10 to 15 years has been mind-boggling. The introduction of computer modeling, computer science, and now AI, in addition to our vast understanding of molecular biology and immunology because of data management, because of data mining, because of bioinformatics, this combination has led to major advances in science. Many companies like ours, which are now developing very cutting-edge therapeutics that we did not have before, benefited from that advantages. So I would see that in the next 5 to 10 years, they're going to be another revolution in immunotherapy, include similar or even more than the one that we've seen 10 or to 12 years ago, that would lead to very, very noticeable enhancement in outcome of patients with cancer. Critical to that is that we maintain our discoveries and we maintain our at least speed of that path forward, which means it requires proper funding, it requires proper structure, and we need to maintain that advantage that we have to be able to do that. But I'm highly, highly optimistic about what the next 5 to 10 years is going to be bringing for cancer patients based on what we know today.
Sami, I have time for only one last question, but address it to our audience. We have this highly educated and interested audience in what you're saying. Tell them, what should they expect in the next 5 to 10 years as opposed to 20 in this particular field of immunotherapy?
Yeah, so I would believe, just to follow up on what I was saying, I would believe that we're going to be having plethora of immunotherapeutic drugs coming forward based on what's happening now and the clinical trials that are developing now. And that's going to really lead to enhance the cure rate from what we know now, which is that 15%, into probably, I never give a number because you're going to come back 10 years from now and say you said that. But I would say it's going to be significant enhancement in the number of patients that will be cured.
Sami, I love the optimism to finish with. Thank you so much for joining us today.
Absolutely. Thank you very much. And good to see you, Francis.
Good to see you. And I will be back shortly. Please, to our audience, don't leave us. I'll be back shortly to speak with Dr. Georgina Long.
The promise of ADC research is really being able to treat cancer more effectively and without the toxicities that people traditionally think of. I'm Philip Moquist. I'm an associate research fellow, and I work with the ADC team in Pfizer Oncology. An ADC is something like a targeted missile. You have the antibody, which targets the cancer cell. It delivers the drug, or what's also known as payload, to the cancer cell. It kills the cancer cell, and by being able to target it, it limits the damage to the normal tissue around it. ADCs are antibody-drug conjugates. They're the combination of antibodies and traditional chemotherapies. This combination was initially invented so that you could get the best of both worlds. You could get the targeting of the antibody to the cancer cell, and you could get delivery of the chemotherapy, more specifically to the cancer cell, to get an improved effect. The combination of the two has shown in the clinic really promising results. Antibody-drug conjugates, they're composed of monoclonal antibodies and a cytotoxic chemotherapy. We've combined the two to get the best of both properties. And the way we do this is we covalently attach the drug to the antibody using a linker. And what this linker does is once the ADC reaches the cancer cell and internalizes, it can cleave selectively and release the payload. What we believe this to do is to be able to improve the lives of patients by having less toxicity and more efficacy.
Welcome. My name is Jeff Settleman. I am the Chief Scientific Officer of Pfizer Oncology. I'm very pleased to be here today with medical oncologist Dr. Patricia LoRusso, Director of the Early Phase Clinical Trials Program at the Yale Cancer Center. Today, we're discussing some of the latest scientific advances driving the next wave of breakthroughs in cancer, with a focus on an especially exciting area that's changing the treatment landscape, antibody-drug conjugates, or ADCs. Thank you for joining, Dr. LoRusso.
Thank you very much, and I thank the organizers for putting together such an exciting and timely topic.
As you know, we're at the forefront of a new era in cancer care, helping people with cancer live longer and better lives. Can you tell us a bit about how far we've come in cancer care and what you see as the key challenges we're still facing?
Oh, yes. So I've been practicing a little over 35 years. And when I first started practicing in the field of oncology or cancer treatment, we had very few effective treatments. And cancer diagnosis was dismal. But over the last 35 years, we've made major breakthroughs in treatment of cancer, especially, I believe, in drug therapies, which is my area of interest. But we do have a long way to go. Even though we've had all these advances and we've seen significant improvements in survival and cures in some diseases, we're still challenged with people still dying of this disease, and also with several of the drugs, the side effects that are caused by some of these drugs. But overall, I'm very excited, especially now in the advancements that are moving forward.
Yeah, it's certainly great to see the advances, but you've described some important challenges, and I think it's becoming increasingly clear that there's no one-size-fits-all model for treating cancer, and that there's still a significant need for innovative new treatment options. We've seen a remarkable surge in oncology R&D, which now makes up the largest area of research investment across all of healthcare, with advances being made in the discovery of new targets, new mechanisms of action, and some innovative combinations. At Pfizer, we're taking a multi-modality approach. We're targeting cancer from a few different angles: small molecules, bispecific antibodies, antibodies that engage the immune system, and ADCs. So Dr. LoRusso, when you look across the research landscape, what advances do you think hold the greatest promise for the future of cancer care?
Oh, there are several that hold the greatest promise, but as you said, R&D is one of the leading areas of investment for many of the pharmaceutical companies. It's because I believe that as we've been understanding more about the biology of the disease, we recognize that cancer is not one disease. It's a multitude of many diseases. Even something like colon cancer, there are many different subtypes of this disease. But what I think is amazing is as we understand the biology of the disease better, what we're understanding is more about what targets we need to go after in order to either eradicate, prevent, or slow down the disease process. So I think that precision medicine, which is multitude, as you mentioned, small molecules, which attack certain targets, antibody drug conjugates and monoclonal antibodies that are attacking, as an example, certain receptors on the cancer cell that make it grow. PARP inhibitors, as an example, that are affecting patients with certain and tumor types with certain genetic alterations are just a few of the many examples of the advancements that are coming forward and that have already been developed, improving the overall outcome for patients with cancer.
Sure. And when you reflect back on the early days of ADC development, would you say that the promise of ADCs is now clearly paying off in the clinic?
Oh, absolutely. Back in the olden days when we first started developing ADCs, they were fraught with significant toxicities. The chemistry of ADCs is very complex. We didn't know as much about the chemistry of the antibody drug conjugates to be able to advance them forward. So what I think is very exciting is not only the recognition that we need new targets new antibody drug conjugates to attack new targets that we're identifying, but also that we need to figure out better chemistry as we're doing to deliver better payloads of drugs that are attached to those monoclonal antibodies of the ADC so that we can enhance the tumor cell kill and minimize side effects.
Yeah, I agree. Those are all great opportunities to advance the next generation of ADCs. And at Pfizer, we are evolving next generation ADC molecules. We're working to better understand their potential and to optimize every component of the ADC by improving the ADC design. And we're exploring new treatment combinations with ADCs. And of course, our ultimate goal is to bring the benefits of ADCs to more patients. So Dr. LoRusso, thank you again for joining us today and for sharing your great insights.
Thank you very much. And again, thank the sponsors for putting on this exciting video and topic. Thank you.
We can love this sunburnt country without the sunburn. Our mission is 0 deaths from melanoma.
We ain't done until we cure cancer.
We actually now have developed drugs that in 50% of the case cure what was a death sentence only 10, 15 years ago.
Hello and welcome back to Washington Post Live. For those of you just joining us, I'm Frances Stead Sellers, an associate editor here at the Post. I'm now delighted to be able to welcome Dr. Georgina Long. She is the medical director of Melanoma Institute Australia. Very warm welcome to you, Professor Long.
Thank you for having me, Frances. I'm really pleased to be able to talk about something I'm so passionate about, and that is curing cancer.
Well, I heard that passion in the introductory video and look forward to learning more about it now. But let's start by talking about that sunny outdoors lifestyle that comes with a cost in Australia. You have the highest melanoma rate, I think, in the world. What are you doing? Tell us about how you are hoping to address that extraordinary death rate and bring it right down.
Great question. Melanoma is indeed Australia's cancer. We do have the highest incidence in the world. But not only that, it disproportionately impacts young people. So 20 to 39-year-olds, the number one cancer in that age group in Australia is melanoma. Yes, it's because of our sunny climate. There are different types of melanoma, but skin melanoma is the most common in Australia. And so that's largely caused by UV exposure throughout one's life. And how are we bringing it down? We've already seen that the mortality rate, the death rate from melanoma has decreased. In fact, in most countries around the world where they have a high incidence of melanoma, who have access to drug therapies, particularly immune therapies, checkpoint inhibitors, we are seeing the mortality rate in melanoma drop. However, in contrast, the incidence is still rising in places like Europe and the US. In Australia, it's starting to plateau because of primary prevention.
So you're talking about sunscreens and hats, those sorts of things that we could all, and long sleeves, all those things we could be doing to protect ourselves.
Absolutely. Clothing, hats, shade, sunglasses, and then sunscreen for the bits that you can't cover up. In Australia, we try to have our outdoor sports training programs for our kids at either end of the day. Just be aware of the UV index and avoid the shade. It doesn't mean don't get outdoors. We love our sunburnt country here in Australia. It's just doing it smartly.
Right. Right. So let's go from that, those primary preventive things we can do, to your work, your research. And you have found that combining immunotherapy drugs before surgery, I think, has a beneficial effect. Can you talk us through that?
Absolutely. So that concept is called neoadjuvant treatment. We know that there are 4 stages in cancer in general. Stage 1, 2, and 3 are what we call early cancer in melanoma, early melanoma. Stage 4, advanced cancer. Advanced cancer is the big killer. And in melanoma, we're even curing 50% in the advanced stage. Now what we're doing is using those immune treatments in stage 3, early, before surgery. That's neoadjuvant. Neoadjuvant's not a new concept. In fact, in breast cancer, esophageal or cancer of the food pipe, we know that neoadjuvant can make the surgery less. But in melanoma, we use neoadjuvant immunotherapy, not for anatomical preservation, but because we see an improved outcome for patients compared with doing surgery first and then using the immunotherapy afterwards. Now, we know that works better than doing nothing, but giving the immunotherapy first, it really improves outcomes, survivals.
So I need a little more help on understanding this. We heard in the previous conversation that immunotherapy helps just 15% of the population. Is that true in your field of melanoma research, or are you seeing something different?
We're seeing something different. When I started in melanoma, it was dismal, absolutely dismal. It was worse than pancreatic cancer, worse than glioblastoma. The median survival was about 6 to 9 months. 25% of people would get to 12 months. This is advanced melanoma, and less than 5% to 5 years. We're now seeing between 50 and almost 60% getting to 5 years, the melanoma-specific survival. That's in advanced melanoma. And we've added on top of that now using neoadjuvant therapy. But what I'm most excited about, immunotherapy, and it's specifically when we use the word immunotherapy, we mean a whole heap of different drug treatments. There's checkpoint inhibitors, which were the first to really make a dent. We have to remember in melanoma that we knew this cancer had a relationship with the immune system decades before. We knew that because IL-2, TILS therapy, the early version of tumor-infiltrating lymphocytes being reinfused into the patient, could work in a small subset. So we knew melanoma had a relationship with the immune system, but it wasn't working for everybody. And it was not until the checkpoint inhibitors over a decade ago, well over a decade ago, when we started developing them. In fact, anti-CTLA-4 was the first, and that was started in 2003, 2004. when they really took hold and we got anti-PD-1, we saw these incredible cure rates. So across cancer, yes, the percentage would be around that 20%. In melanoma, it's above 50% that we're curing in advanced cancer. And this is talking advanced cancer. We're pushing that up. And this is what I'm really excited about, is twofold. First of all, using immune treatment early, when you still have the cancer on board, because we know it trains the immune system better. And I'm talking there specifically about checkpoint inhibitors. That is pushing up that 50% by getting early and training the immune system early before surgery. Patients love it. 6 weeks, 2 doses of combined immunotherapy. We see 60% of melanoma patients have a major pathological response. Our field in melanoma has also refined pathological response. In the olden days, for breast cancer, for esophageal cancer, it was just whether you had a complete pathological response and everybody else. Now, in melanoma, with immune treatments specifically, not chemotherapy before surgery, but checkpoint inhibitors before surgery, we've refined the pathological response so that we understand the immune system and have major pathological response, partial pathological response, and no pathological response. Patients love it because all of a sudden they walk into your room thinking, I've got a 50% chance of this coming back after surgery. They have drug before, they get it cut out. You're able to tell them you've had a major pathological response. You've got less than 5% of this ever being a problem again. If you haven't had a good response, you can change it up. And that's power. Information is power for the patient.
Wow. Absolutely, information is power.
But
at some point, you said, I want to ask you, you set a goal of achieving 0 deaths from melanoma by 2030. That's only 5 years ago. How close are you to that target?
Look, we always set goals high because that's how you get close to it. So I think we're going to get close to it in our lifetime. Definitely, we're going to inch much closer to that 100% than being down closer to our 50% by 2030. And this is what I'm really excited about. So this is the next step beyond the neoadjuvant immunotherapy before surgery. What we learn in melanoma, we can apply to other cancers and vice versa. We need to be sharing more of our knowledge, and we are lucky in melanoma that we didn't have legacy drugs, so that we're able to embrace immunotherapy and novel treatments very well and develop them very quickly. The neoadjuvant platform also enables us to develop drugs very, very quickly because you can get your pathological readout within 6 weeks, and that correlates beautifully with the recurrence-free survival and the overall survival. We've published on that. So it's a great drug development platform. But where I am focused now is what I call the third space. So these are patients who do not respond to single agent checkpoint inhibitor, nor do they respond to double combination checkpoint inhibitor. They are not very immunogenic. They die very quickly, these patients with melanoma, and they represent many other patients in other different types of cancers that do not have those immunogenic cancers. We're not seeing a clue that the immune system is engaged against the cancer. So my total focus and my total goal, crystal clear, is to target that third space, those patients who are still dying from melanoma, and applying what we learned there to other cancers and vice versa. So I have an incredibly, yes, go ahead.
I was just going to ask you about a colleague of yours, Dr. Richard Scalia, who I believe has one of those very aggressive forms of brain cancer. And you were able to take some of the work you've done, I believe, in melanoma and transfer that to glioblastoma in his case. Tell us about that.
That's a great example of the third space. And I have a lot of patients in the third space. Richard's just one example of trying to apply that thinking. But I see about 80 to 100 patients a week with melanoma. And you can imagine 50% of them are in the third space. So I'm constantly thinking, and I also have a translational lab at Melanoma Institute Australia and the University of Sydney, where we're looking at those third space patients. So how I'm applying that, first of all, in melanoma, is we're now using biomarkers to identify the third space early. That's the key. You've got to get novel drug therapies earlier, particularly immunotherapies, earlier in the course of the cancer journey to really get a good signal and to understand how well these drugs work. We're going for cure. Melanoma is now one of those solid tumors where we're not just going for progressing survival, we actually want to cure patients. And that's what I want to do in the third space. So when it came to glioblastoma, the first thing when Richard told me from Poland that he'd had a seizure, Richard's a pathologist. He understands diagnosis of melanoma. He understands cancer under the microscope. He doesn't develop drugs, and he was fraught. He had no hope. And being a medical oncologist and someone who is trying to work in that third space and develop new therapies in that third space, I have a large program of clinical trials targeting many different ways in the immune system to get at that third space, and immunotherapy, particularly checkpoint inhibitor resistance, with some success with some novel drugs I'm working with. But I just applied the thinking, what would I want to do for one of my melanoma patients in that third space? And I applied that thinking to Richard's tumor. He was devastated. He was mute with grief, and the tumor was affecting him. It was in the left temporal lobe. And so when he arrived back to Poland, to be able to present to him this concept of, why don't we try neoadjuvant therapy? Yes, you've been a pathologist and seen, melanoma and diagnosed it, and see what we do with all our drug development program. Would you want to consent to this? And he did want to consent to that. He gave him hope. And that's what I see in my clinic every day. People who are facing terminal illnesses, hope is key. That's what we see in our lab. Hope keeps us going. We've failed experiment. Let's keep looking. What are the resistance mechanisms?
So I want to ask you a little bit about precision or personalized medicine, its costs, and how you take these very specialized treatments and think of them more broadly to address a wide population who may not have the money or access to specialized care that you offer. Are we at risk of increasing health inequities at the same time as we make progress?
I think that is one of the fundamental things that happens when you're making progress. Initially, it's costly and difficult, but in time, it becomes much more available. Let's take the genome. I mean, it's a classic. In 2001, to understand the human genome took thousands and thousands, in fact, millions of dollars. Now you can get your full genome for about $1,000 if you'd like to. And the turnaround time shorter. So things have become cheaper, and that's all technology and technology driven. The same thing with mRNA. That leap in technology, being able to code and synthesize proteins by coding some mRNA, which is now stable because that was the leap, and then being able to inject that into humans to be able to make vaccines against infections so rapidly, and now to make drugs using mRNA that are produced and synthesized in vitro in the human. So you inject the mRNA and then they synthesize the protein which can fight the cancer. That's a massive leap. And with each leap, there may initially be some difficulties to make things widespread, but with time and technology, they become cheaper and much more able for other people to access. So we can't hold progress back, because progress will eventually get rid of these inequities, if that makes sense, because we develop better technologies.
Absolute sense. I also want to jump back on that phrase. You talked about hope. You talked about the power of hope for people who are suffering from cancer. And I'd love it if you could address the experience, what you're seeing in patients' experience, not just in survival rates, but in quality of life?
It's unbelievable. In fact, we've done some pilot studies on quality of life in terms of what are the needs of survivors, because there's now a survivorship issue, which is an excellent problem to have. When patients with melanoma get to 2 years without any progression of the melanoma and a great response to their treatment, which is now the vast majority, over 50% of advanced melanoma, and now moving into earlier stage, we're seeing that in stage 3 melanoma. Patients who know within 6 weeks, 2 doses of immunotherapy with stage 3, and then their pathological response, they know they're cured. So we've now got these survivorship issues. But the interesting thing is the issues disappear by about 2 years after their initial diagnosis of advanced or high risk early melanoma now. They actually don't have any needs. They're back working or volunteering, living a full community life. Remember, melanoma disproportionately impacts the young. So I have quite a few patients in their 30s and 40s having children. In fact, one of my fantastic patients just recently, Avril, she won't mind me sharing a name. She literally at 33 was diagnosed with stage 3, very high risk melanoma, a melanoma that has a 50 to 60% risk of recurrence. Those sort of patients 20 years ago were told, get things in order, don't have children, you may not be around. And she literally took 6 months off, got her neoadjuvant treatment, had a complete pathological response, waited the 5 to 6 months, and then had another baby. It is unbelievable. Like, it's just a blip in her life. When I started in melanoma, that's not what it was like. These people couldn't, their lives stopped at that diagnosis. They literally stopped, and that became their whole life. So it is an absolute game changer. But what I'm most excited about is the technology and the drugs for the third space. That's where I focus all my attention. The people who are still dying from melanoma. And we can apply that to other cancers to try to understand how we can cure other cancers. In melanoma, checkpoints worked. It was then applied to lung cancer, kidney cancer, head and neck cancer. The list goes on. And yes, overall, we're only curing about 20% of advanced cancer. But if we can focus on the third space in melanoma, I know if we can solve that problem, we will unlock so much for so many other cancers. And that's what I'm really excited about. I also don't think we're optimizing our current immunotherapies. And as someone who's been using them solidly for nearly 15 years, that's my career. And I have to say, when I first started in melanoma, I really only saw a patient's face, a single patient's face, a few times in my waiting room because they didn't survive. Now we've got survivors, but I don't think we're optimizing checkpoint inhibitors across cancer, across the oncology field. And I think, and this is a challenge to the oncology field, that we could be curing another 30% across the oncology field just by using checkpoint inhibitors in combination well now. It's not a treatment like chemo where you have to give it every few weeks. You have to individualize it to the individual's immune system. And that's where we are going in melanoma. The third space and personalizing current treatments to maximize that cure rate.
And Professor Long, we have to wrap, but I'm going to give you one last question, but we need to finish it quickly. Address your patients now. What would you say to the next person coming in with melanoma about what lies ahead? It's obviously going to be different from patient to patient. But
it is going to be individualized. I always individualize it, but my one goal is to give people hope. Honesty, hope, planning, but hope, because that enables people to move forward and spend the time they have with their family. And hope is so critical for everything we do, including research and science. And just the world. So my one thing for patients is you have options. We're might start with a trial. Clinical trials are critical. Participating in research is critical. That's why we've got the benefit of these drugs that are available widely, is because people who have come before us have participated in research. And it's the one human thing that we can all do is good, solid thinking and research that tries to answer difficult questions. And don't bite too much from the big hamburger. One step at a time, we'll get there.
What a great message. Hope and join those clinical trials. Professor Long, thank you so much for joining us today.
Thank you, Francis. It's a pleasure.
And thank you everyone else for joining us today. If you would like to hear more of these great conversations, please sign up for a Washington Post subscription. You can get a free trial at washingtonpost.com/live. That's washingtonpost.com/live. Thanks again. I'm Frances Steed Sellers.